Fashion and fitness. These are by far two of the most trending lifestyles in Monterrey, Mexico. Often the two are combined because who doesn't like to look good while exercising? It's a fun way to express yourself with your body and your clothing. The routine is very simple. Buy, use, dispose. Buy, use, dispose. Buy, use, dispose. Dispose, dispose.
Wait, we don't think that's the way things should go. Despite the goodwill our city shows to have a better lifestyle, having an outstanding 65% of the young adult population being physically active, this practice has easily merged with a more serious social and environmental problem: fast fashion.
Fast fashion revolves around the mass production of clothing worldwide, and it represents several significant problems related to our community and environment. As reported by several experts, the fashion industry is a major source of pollution, not only for the use of vast amounts of natural resources needed for mass production, but also for the use of chemicals and synthetic fibers like polyester and nylon.
Polyesters such as PTT and PVT as well as nylon are resistant to natural degradation, and in the context of fast fashion creates tons and tons of waste that won't go away for a very long time. However, we believe synthetic biology can speed up the process. It is very important to take action against the negative impact of fast fashion. That's why in PCB UANL we propose Fiber Eater.
Fiber Eater uses synthetic biology and genetic engineering principles to decompose polyesters and nylon present on sportswear using recombinant enzymes in Escherichia coli. We believe that in this process we can help both our environment and community, providing new ways to mitigate this vast problem that this kind of pollution represents, and to also raise awareness to the problem that is fast fashion. Stick around to see what the team will accomplish this year, but for now, it's time to work.
Thank you. Now, the judges are free to start asking questions for the teams. Please raise your hand so we can proceed with the questions.
I forgot what is the hand. I have used Zoom so much that I forgot.
No problem.
In
Please go ahead.
reactions?
Yes, in reaction.
Okay, I have, welcome team. I used to live in Monterrey, Nuevo León, so I'm very happy to meet Gente Regia here. I have two questions. I am not a biologist or biochemist, so just for you to understand where my questions are coming from. The first question is about, you are aiming to produce, to modify this bacteria to produce 4 enzymes. And I wonder how feasible or difficult is this versus having, modifying, for example, 4 different bacteria for producing each one enzyme and then use all them together in the same bioreactor. This is the first question. And the second is that I am missing an idea about how you would apply this technology or this solution. So it is clear the problem. In fact, I love how you describe your process about analyzing the problem, looking for the most important pollutants. I think this is brilliant. This gives peace of mind to everyone. But I don't see where you would adopt your technology in a treatment plan, in a wastewater plan, how this, what would be the form of the solution? Those are my 2 questions. Thank you.
I think I can answer the first question. Just reminding, it's about why we chose to put the 4 enzymes together instead of individual enzymes in each bacteria. Well, our process of reasoning was that, when we were researching about the problem, we were, as you mentioned, searching for the most important pollutants in your clothes. And what we found is that almost nobody reports which chemicals they use on clothing. So we thought that putting all these enzymes together will actually be like a general solution to all of the problem. I mean, having all the enzymes together can process every single type of cloth in the same system.
instead of, well, you also mentioned that maybe we can put like different bacteria in the same bioreactor. And we kind of did not look at that pathway because we assumed that having, well, different bacteria, even though if it can be like the same E. coli, it will have to like represent having to balance all the parameters. And when we're putting like the 4 enzymes together, we can like think about those parameters that the bacteria need to grow like together, and also like to have the enzymes having their functionality at the same parameters, instead of just like, oh, this enzyme will work at these conditions, this other one at these other conditions. And when we put them together in one bacteria, we have to like balance them first. Well, yes, first before actually putting them all together, instead of like just like an afterthought.
Yeah, very quickly, Jared. In some of the charts where you measure the expression of enzymes, I could see that from the 4 that you had, one is like the champion, the enzyme that dominates the production, and the other 4 are more shy, or they don't develop as much as the other one. This is where my question comes from. Why don't having different bacteria with each one enzyme? But okay, thank you. And what about the second question?
Well, I don't know if any other team member wants to answer it. If not, I could take it.
I can answer this. Our idea, or like what we were kind of planning, is it can be in the factories of clothing. We want a biocircular economy where the textile producer, they can not just produce, but also like recollect and reuse, give a correct treatment to the waste of the textiles. And yeah, because it's like a problem, like the way the companies treat all the residues of the textiles. It's affecting the environment, the economy, and everything. So in this way, it can not just give more jobs, it can also help the environment, and also like the economy.
Thank you.
Okay, do we have any other questions from the judges? Please go ahead, Lorena.
Thank you. Thank you to the team for the video. First of all, I would like to congratulate you on the development of your project. We can really see a tremendous effort in the elaboration of it. So again, congratulations. My question is mostly focused on the biological design of your proposal. You comment that you intend to use 3 plasmids, PSB3K3, for the expression of these 3 enzymes in E. coli. Why did you choose these plasmids? Does it present any particular characteristic? That's my question.
I think I will answer this one too. It's because of 2 reasons. First of all, we as a team have experience with that plasmid, and also with a more technical answer, this plasmid can allow us to have a multiple copies of the biological product that we want. So it will be like easier to produce it while in the lab. But once we have like enough DNA samples of the extracted DNA of our circuits, we can transfer it to other plasmids in order to like test them, or like stabilize them, characterize the enzymes, and other type of experiments that we can do in the future.
Okay, thank you. Get it.
Emiliano?
Yes, thank you. So, well, first of all, congratulations to everyone. A really good project that you developed. I have two main questions, also related a bit to the part of the implementation. So the first one is if you're considering any like pre-treatment to the clothing, because sometimes, especially in sports clothes, there will be these blends that are sometimes, I don't know, 50% polyester, 50% nylon, and there may be a challenge in there in separating that. I know you consider like having like two different like tanks, one for the polyester nylon, or is there something like you will need to consider previously to do that? And the second one is related to, like you're saying that you're proposing to implement like the bacteria to degrade these polymers. But did you consider all creating like a sort of cell-free system, or only using the enzymes instead of the whole organism? Or I mean, what if you consider that? Why do you went for the whole bacteria?
Hello, I think I can answer the first question. If I don't remember well, we propose to make two pre-treatments. One is heat pre-treatment, and the other is physical pre-treatment to reduce all of, we can say, noise of the polyester and obtain that the material that we gonna to degrade, and well, that is the purpose of the project.
Regarding the second question, I also think that a cell-free system will be like more useful. However, when developing the project, we thought that I mean, before having a cell-free system, we needed to like standardize of our full process, and having like the whole system within our bacteria will allow us to yes, have control about all the conditions, and have a more have more impact with the proposal, because when we were talking about our project to our colleagues on our school and other like friends and families, they will always like get fascinated, and they wanted to learn more about how we can do this stuff with a simple bacteria. So yeah, I think that having the system in the bacteria for this like starter phase is most useful. However, a cell-free system will also be like very useful in the future, especially when we try to like expand the project, and yes, well, yeah, I think that answers the question.
Thank you.
Hello, do you hear me well?
Yes.
Yeah, so you're using diverse genetic components to make sure your bacteria does the work you are willing to. So how would you address potential challenges like in the future, like possible mutations, genetic instability that may happen? That's the first question. And the second, will you have byproducts after the process? What will you do with that? Okay.
I think I can respond the second question. The reaction of the enzymes is going to produce certain components, and that may be a little bit toxic if they are in very high amounts. What we do is a toxicological analysis of every compound. So we develop a way of treating it, so we can reuse it, like decontaminate it, make it like in a non-harmful way, so it's not affecting what we have.
Regarding the first question, I think it has to do a little bit more with like the implementation phase of the project, because as Alejandro said earlier in one question, what we refer to with our like bioeconomy is that we will be able to like give our product to certain clothing companies. They can promote like to their customers to bring back their clothes they're using. And with that process, we can also have like quality assessments within those companies, so that we can have full control of, as you said, the genetic components that we're using. You know, having the test with several time spans that can like show us how our project is doing, how the enzymes are working, if there are any efficiency issues, and respond accordingly to that. If we like, well, as another judge mentioned earlier, the curves of the activity of the enzymes can also vary with time, and we can adapt the project according to the necessities of the enzyme activities and the genetic components. The mutations, we will have to like assess them doing, I don't want to say like doing the project again, but we will have like this strong base that we can like modify accordingly to the project necessity in the future. Yes.
Thank you.
Now I see the hand. I have two more questions. I have the questions always in pairs, in the 2 in 2. I would like to know, what do you consider has been the most challenging part in the design of this solution? I understand that you might have a combination of existing knowledge that you leverage, plus also your own development of some knowledge and new approaches and ideas to come up with this solution. So I would like to know what has been the most complex part so far. And then now imagine that you get the funding to say, look, let's do a pilot with a company in Monterrey that produces garment or clothing, and they would like to test this technology in their plant. What do you imagine could be the most complex part of the piloting of this? Those are the two questions.
I can respond the second one again, the second question. About making it in a company, it's a very complex thing in the way of the regulations, laws, because of the treatment at the moment. It is very easy for the companies to pollute instead of giving a good treatment to the textiles. There are a lot of laws in Mexico. There are 5 main laws that are based on ecology, and that can be related to the textile waste and stuff. But each one has different gaps. So it makes very easy to the companies to avoid, for example, the fees, like the penalty fees for the pollution they produce. So it could be easier for the companies to keep what they have at the moment than giving it a better one at the moment, a better option. What we plan to do actually is to reach different companies to make like a round table to talk with them about the correct treatment of the textile waste, because also they have needs. They have problems with the money they are losing, with the pollution, with a lot of stuff. So it can also be good for them. But I think the hardest part of it will be to agree with a company to try something new. If they are like very comfortable at the moment with the polluting thing.
Let me react here, Alejandro. This is the same scenario with carbon dioxide sequestration. There are new technologies to sequester carbon or other greenhouse gases, but in the absence of regulation or enforcement, they just emit, right? Imagine the cement industry. But my question was more targeting the technology side of things. So let's imagine you will find a company with which is conscious about the environment because they are very much attached to the population of Nuevo León. Let's imagine you have the company. I wanted to know what is for you the most complex part of moving from the theory or the design to the real pilot implementation of this solution. You envision challenges in terms of scale, that one thing is to produce 5 milliliters of this, but the other is going to 500 liters. I don't know what is the most complex thing you imagine. And maybe for different team members, there are different challenges. It may happen. I would understand this too.
Oh, I think that when implementing the project in like the real world, I think the most difficult challenge will actually be like how to all the pre-treatment stuff, because as you said, there are some parts of the clothing that need like only 5 millimeters of the product and only other that need like so much more. So as you said, the scaling, bringing the clothes to the system, like tearing them apart will be like a challenge stuff, because when we were developing the project, we only thought like, oh, well, it's already in the system. So like that part to bring it in, it will also be very difficult. Also, like the social awareness of the population about this project. I mean, we mentioned that the stuff that we can do working with other companies, but it needs a lot of diffusion for the general public to say, oh, well, maybe I can bring my closer, because especially here in Monterrey, there are always like a mercadito or something like that, where people sells clothes for secondhand, and I think that also is really good, but it's as we mentioned in our project, it is not enough. So for the public to know about this is, I think it will be very difficult. And regarding our most challenging part while developing, I think it was also like that part, the implementing part, because as I just mentioned, we only envision it within the system and not the process of bringing it in. So yeah.
Understood.
Hi, we have time for one more question. The judges.
Well, my first question is still pending. What was the most complex of the design of the solution?
In the beginning of the design of this project, one of the hardest challenge for me was the selection of the enzymes, because the polyesters that we study, there are three: PET or PET, PTT, and nylon. So PET or PET, that is well studied. PTT and nylon, there is no much studies about it, and of enzymes that can degrade these polyesters, and that led to also a hard work of research and select the perfect enzymes to degrade these two polyesters. And well, that is for me one of hardest challenges of all the design of this project.
Thank you. Thank you, team, and our judges for their questions and comments. We have now concluding this judging session. For the students, please exit the room, and we will have a 5-minute break before we continue with the next team. I will now start letting the teams in 1 minute. Welcome everyone to judging session number 6. We're about to start by sharing the team promotional video. After the promotional video, you will have up to 22 minutes to answer any questions from the judges. Remember to be brief, clear, and concise. Only student members are allowed to answer the judges' questions. So I'll start by sharing this team's promotional video.
Xochimilco del Náhuatl, campo de flores. Representa cultura, tradiciones y colores. Las flores, protagonistas de este lugar, no siempre benefician al ecosistema. El lirio acuático en exceso provoca eutrofización en los cuerpos de agua. En consecuencia, las especies que coexisten en el ecosistema se ven afectadas.
En 2023, IOM One propone hacer una modificación genética en Streptomyces lividans para producir PHA, utilizando como sustrato Eichhornia crecipes. En otras palabras, producir un plástico de origen natural de fácil degradación utilizando los residuos del lirio acuático.
Lo cual podría reemplazar el plástico convencional minimizando los residuos. Recordemos que Xochimilco es de todos y todos podemos cuidarlo.
Thank you. Now, the judges are allowed to ask questions to the team.
Hello. So for me, it's not very clear. You will use the lily plant as the sole substrate. Will you not add like salts or any other substrate to improve the biomass production of the bacteria, or how would you do?
We pretend to add other things like buffers or something like that, or the organism could grow. But in general, we try to, the main substrate is cellulose from aquatic lily.
Go ahead, Diana.
Yes, as my classmate mentioned, we decide to use the biomass from the water hyacinth for produce the PHB because this plant is an invasive species in Xochimilco. So we pretend to resolve this problem using this biomass in excess for produce PHB. But technically, the chassis could degrade another cellulose from other plants.
Can I ask?
Please go ahead.
Yes, thank you. Well, first, congratulations to the team. I love your video, your promotional video, because it shows Xochimilco, the people, and the plants, the lilia. I think it's beautiful, and I love this place. I've been there a few times, and I love it. I have a doubt. I don't understand well something, and I would like you to try to explain a bit deeper. You start saying that there is an issue about eutrophication, okay? And then you start saying, well, we are going to modify genetically this plant. But then soon after you talk about using what you can do with this plant to produce cellulose and do a biomaterial, right? But what is not clear to me is when or where in the process you are going to fix the issue of the eutrophication. Is it automatic? How this work?
Well, first, sorry because of I couldn't put the background. It didn't allow me. And well, the problem with eutrophication is that, you know, the water lilies actually cover a huge part of the light, and it actually allows for bacteria proliferation, which generates this kind of algae. At first, we thought that we might use synthetic biology in order to reduce totally the amount of water lilies, but we realized that it was actually that they had some biological and ecological function in Xochimilco. Like a lot of the agriculture workers use it as a abono, and also that a lot of native species depend on it, like some ajolotes still put their eggs in the water lilies, and some other species use it as a food material. So we don't want to end completely the water lily because it's now part of the ecosystem, want it or not. It's still a plague though. So what we want is to reduce the amount, so it's in control. And we want to produce this bioplastic as a mean to reduce it in a sustainable way and in order to offer the community an economic activity.
Okay, now I understand much better. Thank you. Thank you. Okay, and then the whole project is about how you do that. But then how, I like this approach to say there is too much water lilies. Let's take some, and taking some, we transform this some into something else that has value, and we reduce the amount of water lilies, so we reduce the pressure on the lake. But how you will know how much lilies you will take away? It's something that you have like I don't know, a percentage of something, how you measure the concentration of something. It will be something visually. How this would happen?
Well, I think it could be visually because the aquatic lily grows really fast, so we can clean the whole lake, and in a few days, again will be full of aquatic lily, so it could be visually, and right, I think that could work.
Oh, excellent. That's fine. Ricardo, I don't want to. No, now it's Maria Elisa. No, yeah, sorry. I have a question for Ricardo. Ricardo, can I give them an idea for them to think in the future?
We can wait until the end for the comments. For now, let's continue with the judges asking the questions.
Okay.
Thank you. Emiliano? Or Maria, are you answering the previous question? Go ahead.
Lo diré en español y me ayudarán a traducir. Lo que haremos con el lirio para saber cómo haremos para saber qué cantidad. Lo que se planea hacer es hacer un estudio topográfico y también con modelado matemático, que pues en base a esto con entrevistas e ir justamente a Xochimilco y hacer este análisis, es cómo vamos a saber qué cantidad de lirio acuático se va a utilizar pues justamente para este estudio, solamente que este paso va a ser para la siguiente fase, porque es un estudio muy largo y muy complejo, pero todavía uno pues de nuestros asesores es quien nos va a ayudar, ¿no? Entonces tendríamos que ir a hacer ese trabajo en campo. No sé si me di a entender.
Well, she says that we plan to do a topographical study and a field trip for the next stage, because right now we don't have the means, and we also plan to ask help for the community that it's already there, and also we plan to do kind of like a census, to know which, because the water lilies are measured by area more than biomass, because of how much part of the lake they occupy, and that's how we want to measure in part, and we hope to get the resources for the next stage.
Thank you. Emiliano?
Yes, well, first of all, congratulations. I really like your project and proposal. So I have two kind of more technical questions. So the first one will be like, if you are considering any pre-treatments to the water lily, considering, that sometimes when you use agricultural resources or things that are considered like a waste, you don't have like an standardization in terms of, I don't know, the amount of cellulose or stuff. So if you're planning to do any type of pre-treatment to the water lilies before introducing them as a substrate. And the second one will be, how are you planning to measure the cellulose and hemicellulose degradation that will happen in your bacteria?
Can you repeat the question, please, more slowly?
Yeah, sure. Sorry. So, once you collect the water lily, what do you plan to do to the plants so that you can get an standardization? So, let's say your bacteria needs, I don't know, just to say some number, 50 grams of cellulose. Like what steps are you considering for you to have a standard once you collect the water lilies? I don't know if there's a process or something you're planning to put in there.
Yes, that was an important part of our design, and for that we was discussing a lot of ideas about it. But in this process to make it real, this design, first we was thinking in recollect the water hazard, and then put it in a process of purification with water, and take it the steam, because in this part of the plant there is the percentage appropriate to take the cellulose. All through our design, we have some problems. So for that, we decide to make the assembles, the genetic circuits, only using cellulose. And we design the genetic circuits for degrade only cellulose, because for degrading hemicellulose, we need more enzymes. So for this part of the competition, we only design for to the objective of the great cellulose. But all this is in the proposal and it is explained better. And then we purpose of use an bioreactor for the production of the PHB.
For the students, feel free to go ahead and answer the questions. You don't need to raise your
hand. I could talk about the last question.
Yes, please go ahead.
Okay, thank you. The second stage of mathematical modeling works to give us an idea of how to CRISPR design and is a variable for the PHB production. And the results obtained were in growth rates, and we're divided into 5 stages. And the first to observe the connective of biomass, which in this case Cupraeus necator, and the production of PHB in the model. And the third stage tell us that at least 0.1 microorganisms will obtain 43 of PHB, and the fourth stage and 5 are designed to demonstrate and compare use to CRISPR interference gene PTSN with PHAC, and demonstrating that it is better not to have to go working simple CRISPR genes and thus stop our organisms if we to take it to a bioreactor in the experimental phase, and to a certain point does not grow because it stores it as food and just accumulate.
Thank you. Gabriela?
What is the media or the time that you're taking into account into transforming the lily water into the bioplastic? What's the media of the time you calculated that?
Do you have an answer?
I think right now we we don't know exactly how much time, because we pretend to wait for the experimental part. We only, but we're in the recollecting process. I think the process starts there. So in that part, we could take a few days in the recollect and also in the cleaning, and after in the process that Diana said. And after into the transformation, we don't have an exactly idea, or we want to wait for the experimental part.
That's actually a good question, and we are going to take it into account in order to improve our project. But I guess we might, well, according to the model, we might need around an hour.
An hour for one lily plant, or for how much? Or is for the bioreactor per volume?
Wait a minute, please. Let me remember, please.
That you say 1 hour maybe is for all the bioreactors, so you may calculate by volume.
Well, for wait a minute. Well, according to some literature, per 0.1 unit, let's say, of the microorganism, we might get 43 units of PHB, according to the growth rates.
Okay, thank you.
Yes, Rafael?
Yes, I would like to know more about how you engage with the community of Xochimilco, the people that live working there along the development of this project. And I'm asking this for many reasons. First, they live there; it's their main of life, main of living. And also because socially in the world right now, when people read about genetic something, whether it's GMO or not, people sometimes feel in panic. So what was the involvement with them, and how familiar are they, or some of them, with your approach?
Bueno, la relación con la comunidad de Xochimilco fue un acercamiento en donde se buscó una interacción y tener una empatía con la comunidad. Fue nuestro primer acercamiento y dentro de esto se trabajó la psicología social y ambiental. Entonces lo que se buscó fue conocer las necesidades de la comunidad precisamente para saber de qué manera vamos a trabajar y implementar estas estrategias para Human Practice. A partir de estas entrevistas y de esta relación con la comunidad fue acercarnos y preguntarles cuáles son sus necesidades, preguntarles cuál es su día a día y desde ahí ellos nos empezaron a darse confianza y eso fue lo que nos ayudó mucho a comunicarnos con ellos y ellos pues han sido muy amables y han sido muy participativos dentro de nuestra investigación. Entonces, más que nada fue un acercamiento normal, platicar, hablar y todo eso es lo que nos ayudó mucho.
Well, do you want me to translate it, or is it okay like that?
For me, it's okay. It's Ricardo if he wants this to be translated for the YouTube channel.
For now, we just need one more minute for questions. Is there any final questions from the judges? Or Alejandra?
Oh, I want to complement what Eli said, because it's right that here in Mexico, when people hear about genetic modification is demonized, the people get scared when as soon as they hear it. But we try to explain in the easiest way as we could, and I think it works because they got enthusiastic with us, and also they want to participate in our project. And I think we try to teach them that genetic modification is a tool, and if we use correctly, we can do amazing things, and we can improve our lifestyle, and is important in a developing country like us. And I think we they they took the project good.
Thank you. Thank you.
Thank you, team. And thank you to the judges for their questions and comments. We have now concluded this judging session. Please, if you're a part of the team, please exit the room. And we will have a 5-minute break before we continue with the next. Thank you, everyone. Thank you, judges. I'll start letting the following team in right now.
Progress.
Welcome everybody to judging session number 6. We're about to start by sharing your promotional video. After your promotional video, you will have up to 22 minutes to answer any questions from the judges. Remember to be brief, clear, and concise. Student members are the only ones allowed to answer the judges' questions. So the judges, please raise their hands when you have a question, and students, feel free to answer any questions as they come. Thank you.
Hi, Barbie.
Hi, Ija.
You wanna go find some microplastics?
Sure, Ija.
Jump in.
We are Barbie Group in the real world. Microplastic is not fantastic. We must be aware it is in everywhere. In treatment station, it passes through filtration.
Come on, Barbie, let's go find.
We are Barbie Group in the real world. Microplastics, tiny bastards. We must be aware they are everywhere. In treatment stations, they pass through filtration. With a probe made of gold, we measure the value of a plastic drop calling peanuts.
We combine PVPs with the tin cups and nukes. We can catch microplastics in the water.
We think that we can take plenty has magnetic core. We are Barbie Group in the real world. Microplastic is not fantastic. We must be aware it is in everywhere. In treatment station, it passes through filtration.
Come on, scientific, let's go catch it.
Yeah.
Come on, Martin, let's go show it. Come on, HP, let's go solve it.
Yeah.
Come on, Pierre, let's go connect.
Thank you, team. You can now start asking questions, judges. Gabriela, please go ahead.
Hello. So the Barbie system, it implies that the companies will have to modify their infrastructure in order to add the system. So how will you make sure that the companies will be willing to invest this money to actually modify all the infrastructure? And not only regarding the water quality, how will you make sure they will be buy?
Team, feel free to answer.
Tem algum áudio que tá ligado?
We think that microplastic is something. it's really an issue in today's world, and we think that the enterprises are willing to invest this money um, to the world because it has to be sustainable, right? We have our planet and we have to be able to maintain it. And microplastic is an issue that is affecting human health. And if we don't take action in this direction, we can't go anywhere.
Thank you, team. Do we have another question from the judges? Please go ahead.
Yes. Gente, boa tarde. I find the approach, your approach, very original. If I understood it well, you have like a two-process step. First, you have this bacteria that will bind with the microplastics, and then you have this magnetism that will separate this from the rest, and then put the microplastic somewhere before and allow the water to flow. Can you tell me a bit about the origin of the idea? How you came up with this idea about binding microplastics with bacteria?
Well, so we actually based our idea in a previous iGen project that already had used encapsulings and the SpyTag SpyCatcher system, but they did not have the magnetic part. So we searched for alternatives to do the removal of the complex because the other iGen project, I'm sorry, they just incinerate the complex and it's not good for the, it's just not good. So we searched for alternatives and we find that encapsulings can be magnetic. They can have nanoparticles inside it. And then when we found that, we included in our project because it would be a good way to work with it.
Okay, thank you.
Emiliano?
Hello, everyone. First of all, congratulations on your project. So my question is around, legislation and the laws in Brazil. So you talk about throughout your project that there is a lack of regulation for microplastics. So my question will be like, about what you found, what would you propose for the legislation? Do you think there's any interesting that could be done in that manner to solve the problematic, or what insights do you get from that?
Hi, Emiliano. Thank you for your question. That's a very interesting problematic that we faced in our project because we actually talked in human practice with some lawyers and we tried to understand how Brazil sees the microplastics today. And there is a huge lack of legislation and attention to this problem. We actually have legislation for plastic, but not for microplastic. So giving attention, putting this topic on focus, it is very important to give the proper attention and significance to this.
Rafael?
Yeah, I would like to go back to Gabriela's question and answer. Gabriela asked that companies would need to adopt your technology in company, but I would like to clarify whether the company is a water treatment company or is each company that pollutes, and then your technology will help them to reduce this pollution. Who's the client here?
Rafael, the client is the water treatment plant. That's right, and that's very important to know that every alternative we have for treating microplastics, we will have to ask for the company for the water treatment plant to change its infrastructure. So if we try the SynBio approach, it might be the best alternative in every aspect, but it would be the best alternative thinking about the infrastructure of the water treatment plant. That's because we can use what it is already used by several water treatment plants, and we wouldn't have to make a huge difference in the infrastructure by itself. Is
Yeah,
it
you
clear?
will add a new step in the process.
Yeah, we wouldn't have to change the whole water treatment plant, but just a part of it.
Okay.
Emiliano?
Yes, so I have another question regarding to, within your system, does it have a range? Do you need the microplastics to be in a specific size for you to be able to capture it? Considering that the microplastic might vary from very small to particles to some bigger ones. So, does your system work for several types of size for the microplastics, or how are you considering that?
Okay, can everyone hear me? In our system, it's curious because the magnetic system works better with small microplastics, but the capture system works better with big microplastics. So big microplastics can be captured with easier, and because of this, we have more particles and the MPB complexes that has the magnetic parts that can capture the more MPB complex can capture the big microplastics, and it means that the big microplastic has more magnetic parts, and because of this, the magnetic works better with the big microplastic too. It's simple. Small microplastic has small mass, and the magnetic force can take this easier. Big microplastics has more MPB complexes, and then the magnetic force can take it easier too. And yes, the system works. It works very well with different sizes of microplastics.
Thank you.
I'll just like to complement what Gabriel just said, that we developed different docking simulations with different types of plastic, and we not only simulated different plastics but also different size of plastics. So we will work with different types of sizes and types.
Thank you, Rafael.
Yes, in terms of in the future, if you were able to implement your technology in a water treatment plant, Brazil is a big country, is the biggest in Latin America. You imagine that you would start in Campinas, close by Senepem, or you would imagine that maybe Manaus is a place that pollutes a lot and it's on the river in the Amazon, and then you could have more impact in a place like that. What's your view about that?
The whole process was designed to be replicated. So we really think that It is a system, a complex system that can be replicated throughout Brazil and also throughout the entire world.
Yes, I thinking about testing. Let's imagine, okay, we have to start with one implementation. Now, maybe the first is around the corner because you need to work very close to be feasible. But if you were to implement this in Brazil at a bigger scale, do you as a team have like a priority of low-hanging fruits, places that with this technology you could solve massively the issue of pollution?
Yes, so we wanted to try in places that we know that has microplastics, that already has literature describing the microplastics. So we know that we can find microplastics in the water in Rio de Janeiro, and we also can find microplastics in Brasilia, which is the Brazil's capital. And sure, it would be very nice to try this in Manaus, because I'm from there, so I know about the pollution. The rivers there are very polluted, and it would be a great candidate to this. But yes, we would like to expand our project to other cities.
Okay, thank you.
Do we have more questions or comments from the judges?
I have another question. It's in terms of speed of the binding. How fast? I mean, a treatment plant has a throughput, so water comes in and water goes out. And if the process of binding the bacteria with the microplastics is extremely slow, this could reduce the throughput of the treatment plant. Do you have any idea about what's the capacity that you need in terms of speed to really don't create a bottleneck in the plant?
Yes, we have. The math model answered this question because we can relate the time that we need to capture the microplastics based on the concentration of complexes. So if we put a lot of complexes, it will be expensive, but with the time decrease a lot. And we just can find the necessary number of MPB complexes to catch the microplastics in a time enough to be compatible to the existent processes. Basically, we use the rate of the water treatment plant. So it is a data that we can find in literature. So we use the number of complexes necessary to capture the microplastics in this time. And of course, we need more data of experimental data to fit the mathematical model to know exactly the number of MPBA complexes or exactly the time that we need. But I think that the next steps of our project will answer these questions. So the mathematical model, we put a constant that we call it the efficiency, and we don't know what is the efficient without the experiments, but we can estimate this, and based on this we can estimate the time that we will take, and we choose the data, the time that the water treatment plant takes. We
understood. Fortunate.
Exactly. So I'm sorry. This is the time that we need to capture 93% of the microplastics, and it depends on the efficiency. Is eta, the Greek letter.
Mm-hmm. Understood. Thank you.
Please, Gabriela.
Compared with the enzymatic degradation, how much cheaper is your technology?
The enzymatic degradation is a long process, so it would be better to remove the microplastics and then decide what to do than expect to put an enzyme or a microorganism there and wait for the plastic to be degraded. So we expect to this our project to be an advantage when compared to enzymatic degradation, because it takes months to a microorganism or an enzyme, biological enzyme, to do the same similar thing.
Yeah, saving time represents also money. So thank you.
Sorry. So another aspect that I want to talk is that our process can be complementary to the enzymatic degradation. If you think that if you put some enzymes to degrade the plastic on the water treatment plant, first we have a lot of microplastic distributed in a lot of water, and we cannot just put the enzymes to degrade. It's more logic to capture this microplastic, to put this microplastic in another section and more in a biggest concentration, and then we can use some enzymes to degrade. So I think that the two processes are complementary.
Do we have any other comments or questions from the judges? Emiliano?
Yeah, just one final question from my part. So I was seeing you developed a machine learning model. Just want to understand a little bit, if you can tell me a little bit more about, I think you were saying that you didn't have actual data, but you had to complement it with something else. So if you can just guide me a bit through how your model works and what was the source of the data to train it.
Okay, basically the machine learning model is about the sensor, and we take it to the literature. Basically, it's a model that understands the descriptors of the microplastics, and then we use these descriptors to find some properties of microplastics. So as the type of microplastic or the size of microplastics, and these descriptors is get for the electrical measure called impedance. And with the sensor, we can get this data. So this data is so related to have the sensor. And it's a next step of our project is turn it experimental and construct a sensor. And with the sensor, we have the data. And basically the data is we pass a microplastics and a sensor make a measure. And we know these microplastics that to pass. And we do this a lot of times, and we construct a data bank. And with this data bank, we can use the microplastics that you know to predict microplastics that you don't know what is. Basically, is this.
Yeah, we have like reached out a lot of articles in the literature to find microplastic data and to like formulate a proof of concept of the machine learning model. But unfortunately, most of this data is not like open. So we reach out like Matt Carlson, which is a researcher from MIT, and we speak with him to like help us with this model, but it's not possible at that time. So
I would just like to complement what Giovanna and Gabriel just said, that we can label our data in plastics, seeds, organisms, and bubbles that are captured as a signal of the impedance system. And we can identify using a KN method of machine learning. So we can group the data visually and locally in our sample. And
Thank you, team. Thank you, team, and to the judges for their questions and comments. We have now concluded this judging session. We request all of the team members to leave the room, and the judges, we will have a 5-minute break before we continue with the next. Thank you, everyone. I'll be letting the students in 1 more minute. Welcome everyone to judging session number 6. We're about to start by sharing your promotional video. After your promotional video, you will have up to 22 minutes to answer any questions from the judges. Remember to be as brief, clear, and concise as you can. Only student members are allowed to answer the judges' questions, and please feel free to answer whenever you hear the question. I'll start by sharing your promotional video.
Colombia, tierra de aguas mágicas y puras, se encuentra actualmente en un problema invisible pero peligroso. Microplásticos y otros contaminantes ingresan a nuestros recursos acuáticos, afectando así la vida marítima y la salud de las personas, principalmente aquellas que se encuentran con acceso limitado a agua potable. Actualmente, los microplásticos no solamente afectan la calidad visual de nuestra agua, sino que afecta la vida acuática y llegan hasta nuestros platos. Por otro lado, la minería y la industria se han encargado de introducir metales y contaminantes tóxicos como el cadmio, los hidrocarburos y el mercurio.
Teniendo en cuenta esta problemática, nuestra innovadora propuesta junta la vanguardia de la biosintética como una disciplina interdisciplinar. Ofrecemos así un biosensor compuesto por un circuito genético que nosotros mismos diseñamos y recibe como entrada un agua contaminada, retornando así como salida una cuantificación de qué tan contaminada está esta agua, ofreciendo así modularidad para adaptarse a cualquier contaminante, estandarización de la medida y accesibilidad de este económico dispositivo para cualquier población y comunidad.
Hemos desarrollado un sistema genético modular, el cual intenta detectar microplásticos, hidrocarburos y metales pesados. Nuestra solución la dimos primero en un sistema antitoxina. En segundo lugar está la detección cuantificable, en la cual en proporción a una concentración del contaminante se emita un citocromo para que eventualmente Shigella, mediante unos microtúbulos, pueda transportar electrones para que así un sistema de electrodos pueda identificar el potencial eléctrico y así dar una cuantificación.
Únete a nosotros y sé parte del cambio.
Thank you, judges. You can now ask questions.
Okay, I will start, Ricardo. I have 3 questions here. Well, I have many, but I'm going to shoot only 3 questions. Well, first, congratulations for your project. You are trying to solve a really, really important problem. And my questions are, first, I mean, your video of 20 minutes explains a lot of things, but I would like to understand if you are talking to develop 1 biosensor or 4. So you're trying to have 1 biosensor that can detect hydrocarbons, cadmium, mercury, and microplastics, or it's 4 biosensors? This is one question. The second question has to do with the interface of the solution about the quantification. You are envisioning that the quantification is going to be yes, no, like it's contaminated or no, versus very high, high, medium, low, very low. Because depending on your answer for this second question, I will make the third question later on.
Well, first of all, thank you for the congratulations. We are proposing to develop 4 different sensors where all the sensors work pretty much the same. However, the beginning of the generic circuits change depending of the contaminant we are trying to detect. And about the measure, we hope it to be, well, no, we develop a system that has the ability to quantify this.
Yes, but I understood. And thank you for the first answer. And I understand where you're going, but let me go a bit deeper here.
Okay.
So you will have a device, right? You or the user, the end user will have a device. And he will need somehow a mechanism to see the results of the biosensors, right? Might be in colors or with a bar or with a line or with whatever you want. My point about how they will see the results of the biosensor, it's very important about how to read the result. And let's say one is the option is to say it's contaminated, yes or no, which is a very low way to quantify. But in order to get more accuracy, which is desirable, then you have to say it's very high, high, low, very low, or give numbers 0, 1, 2, 3, 4, 5. Are you aiming for can you give me an idea about how the user will see this quantification?
Yeah, sure. The idea is that we will have our bacteria connected to an electrical system that's through an Arduino. And the idea is that the Arduino gets the voltage that's given from the bacteria. And from that, you will be able to measure not just a level of the pollutant or the contaminant in general, but also you could, in a LCD screen, you will be able to see the level and the amount. I mean, for example, grams per liter, or I don't know how to say that in English, but
Say in Spanish if you want.
Partes por millón, por ejemplo.
Uh-huh.
You
Part per
could
million.
measure that, and that's given by the bacteria with the relationship with voltage, but we will do it through an Arduino One and the LCD screen. Also with some LEDs that will indicate to the user which is the level, if it's potable or it's unpotable water.
Also part of the question of that answer, we would like to focus different types of results to the person. For instance, if it's a corporation that knows how to interpret real quantification numbers of ppm or whichever measure of concentration, they will have the exact concentration. Meanwhile, for instance, some agriculture or some person that is that doesn't have the scientific knowledge to interpret those results, we intend to have LEDs that will give a qualitative result about the concentration of the water in terms of how healthy it is or according to their purpose.
Okay. I have more questions, but I prefer that Gabriela and Emiliano shoot questions, and I will go back with my questions. Thank you.
Gabriela?
Hi. Do you think the system sensitivity will change over time?
Will you please repeat the question?
If you think that the system sensitivity will change over time by being exposed to the samples or to the contaminated water for a long time, maybe the system sensitivity will change. Do you think it will happen?
Well, of the way we develop it, the system includes like some medium in order to the bacteria to could live. So we hope the sensitivity and accuracy of the measurement to I don't know, be good enough for all the time that the medium lasts.
Yeah, so, well, first of all, congratulations, everyone. I have my question is related to, do you have any type of containment strategy, for this kit, especially if you're going to use it in, I don't know, a river or something? Let's say someone by accident, drops the kit and it goes into the water. Do you have any containment strategy for the bacteria not to spread? And also, kind of in a follow-up question, do the persons that are going to use this kit need any type of training? And if so, is it a manual? Is it someone that explains to them how to use them? Yeah.
Okay, so regarding to the containment, yes, we propose a system that is the bacteria is inside a small container, and then they are connected to the LV medium to feed the bacteria. And on the other side, there's another container to all the garbage that is thrown by the bacteria. The idea is that initially how we propose the system is not to measure directly from the river, and but the people that is responsible to taking the measurements, that those will be the ones that will be putting the sample into our device. And sorry, could you repeat the other question, please?
Yes. So, what type of training is needed to use the device? Are you considering like putting a user manual or a video, or does someone needs to be trained to use it? What type of knowledge do you need to have to use it?
Yes, in fact, in the Juggle page, there's the user manual that of the device we designed, and there's the step-by-step that is needed to use our device. Also, we have a video that explains all how we could be able to measure with the device. But principle is in the user manual that's in the Juggle page.
Gabriela, sorry.
Compared to other conventional quantification methods, how is your system better or more accurate? How you compare?
I think our value proposition, more than accuracy, is actually having a system that can be used in rural areas and in places where there really isn't an access to complex machinery or laboratory equipment or very specialized people. It's more like an accessibility kind of thing in that's what I would say about that.
On the other hand, on the case of Colombia, we investigated and we consulted with a group that are aqueducts that are from the rural part of Colombia. They don't really have machinery that is really according or corresponding to their own infrastructure, because they use traditional materials to construct their aqueducts. So with this type of device, we intend that is more reliable for them, and we can construct certain parts because we intend to construct more and develop more this project, and having recommendations from them that can be helpful for them. And also, they indeed don't have that much of the monetary resource to make measurements from analytical measurements from analytical chemistry. So this is more affordable for them, and it's more it's best for them to their corresponding infrastructure they manage.
I come back. One of the things I love about the project it's that you are tackling an issue that affects always poor people. I mean, the ones that live in rural communities next to illegal mining or lodging or extractive industries like oil, etc. So I think this is a huge problem, and these communities would be the ones that benefit the most of that. But together with this, you are measuring water. And if for some reason the measure it's not accurate, you can create a big issue, and it's a liability, right? So this brings the concept of metrology. So if you are releasing a device that is going to measure water, in order to create some trust for everyone, for you and for the users and for authorities, you need a certification that the measures of your device are realistic and are accurate and reliable. Do you have in your roadmap the approach to the Superintendencia de Industria y Comercio or any agency that in Colombia can certify your device?
I think we haven't directly included that in our roadmap, but we have thought about the processes that we need to undertake in order to determine like the accuracy of our measurement, and also maybe increase it in case we need more, as you say, like more accuracy in order for it to be useful for the people that we want it to be employed. And I don't know if Anna, you were gonna say something else.
Yes, I want to say something, because the actual process, we search about that to understand where's the main point of our device, and in which part of the whole process will be our device. And we found out that El Instituto de Salud is the responsible of taking these measurements, and they do it through a certain measure that's called IRCA. El IRCA, it's the level in which you can determine if the water in Colombia is potable or is unpotable. So the idea and how we propose the device also is set it in this regulation, in this level of pollutant that's explained in the IRCA. Now, there's an issue that we didn't tackle directly, but we did all the theoretical part that we investigated. Is that there's the regulation that certifies the laboratories, and our device will be in BIMA or will be I don't remember. La DIAN also, yeah.
I know both.
And those two enterprises are the responsibles of certify, and we did that in the theoretic part, but we didn't talk directly to them due to the lack of time. We wanted to do a meeting with them, but we weren't able to do that.
Okay, and how much is the cost of the device? To produce the device, the bill of materials of the device, it's $500, $200, $100.
So to create the device, I will open the Juggle because we have that number there. But we have an operative cost for the user, for the final user. We propose a system somewhat like Netflix, in which we put the device into the community, into the aqueduct, and after 3 months, we go to the aqueduct to check that everything is going well with the device. We also change the bacteria because there's a problem with them, they will die. And we want to do that every 3 months, but we will I don't know that word. We will get a cost of 16,000 pesos. That's not so much in Colombia, so it's affordable regarding to the actual devices that are currently on the market. And I will check in the Juggle because I don't remember exactly what's the cost of production.
Okay, well, you check.
For reference, 16,000 pesos is like $4.
Okay, no.
Like the
But
Colombian.
Understood. Here is the thing. Sensors can be extremely expensive. I mean, the agency that in Colombia is measuring if water is drinkable
maybe each device costs $10,000, $20,000, right? So this brings the point that when you go to the process with INVIMA to measure the accuracy or certify the accuracy of your technology, maybe they detect things that you need to comply or assurances that you need to provide that could increase the cost of your device, I don't know, 10 times. But here is a trade-off. So the cost accuracy. So you may think, look, I just want to say yes or no versus provide a scale of 20 different points. So this could be crucial for the operational of the project, of the solution.
So I'll ask you another question because I got a little confused when you responded like you needed to change like the device every 3 months or so. So, you know, are you thinking of this as a one-time use device? Like you buy it, you test it, and you discard it? Or is it something that you install in some place? You know, like how are you thinking about the usability and you know, how many times like the user can people can use it? You know, like if you buy the hardware and you need to buy replaceables, or is it just a thing that you can buy one time and discard?
Yeah, so the idea is to initially put the device into the aqueducts, not directly from the water, because there is a thing that we didn't work further. That's about the connection with all the aqueduct system that's currently on the aqueducts. So we won't do it directly from the water, but we will have the device in a different part of the aqueduct. But every time the people who need to use it could use it for a maximum time of 3 months, indefinitely, with the infinite number of samples that you would like to measure. After that, after the 3 months, the idea is to go to the aqueduct directly, or to provide a system of sending the new replacements of the main device, that's the bacteria that's in there.
Do the judges have any other questions or comments?
I don't know if I cannot see the name of the lady that now the camera is off, that says Castellanos. The name starts by
Ana.
A. Ana, sorry,
Sofia,
Ana.
sorry.
Ana Sofia, I don't know if you had the chance to look at the cost of the device, the cost of manufacturing the device, not selling it or things like that. I will also look at it in your all the data that you provide. You're mute.
Estás en mute, Ana.
Perdón. If I'm not wrong, I think it's there, but we also did it apart from that, and it's around 80,000 pesos for the production if we do it many devices together.
Could be cheaper. Okay, and 80,000 pesos is like $100.
$80,000 pesos.
$20.
$20.
Yeah, approx. It's like divided by 4.
Okay, you're gonna kill the industry of water measuring. Okay.
We hope so.
Okay.
Okay. By now, we have finalized the judging session for this team. Thank you, team, and thank you, judges. You can now exit the room, team members, and thanks again